The overall goal of this procedure is to describe an acute large-scale neuronal recording from the left and right forelimb somatosensory cortex simultaneously in an anesthetized rat using a silicon probe. This is accomplished by first anesthetizing the animal with urethane and later by placing the rat in the stereotaxic frame to make a 2 to 3 centimeter incision over the skull. The second step is to place the ground and reference screws. Then, 2 craniotomies are made above the somatosensory cortices and the dura mater is removed. The final step is to carefully introduce the silicon probes into the brain and lower them slowly to the desired area. This procedure allows the recording of local field potentials and spiking activity from tens of neurons.
The main advantage of this technique is that silicon probes allows to record from a large number of recording sites. Moreover, multiple recording sites can be arranged over a distance of millimeters, allowing to record simultaneously across multiple cortical columns or across multiple layers in one cortical column. Importantly, geometrically precise distribution of recording sites allows to investigate spatial relations between neuronal populations.
This method can help answer key questions in the neuroscience field, such as how neuronal population activity is involved in memory, plasticity, stimulus coding, decision-making, as well as how these neuronal interactions are affected by different drugs.
To begin this procedure, anesthetize the rat with isoflurane at a concentration of 3 to 4% and maintain at 2% to prevent the animal from getting stressed by injection. Then, inject 7.5 milliliters per kilogram of 20% diluted urethane solution intraperitoneally and divide this injection into 4 applications, each separated by approximately 30-minute intervals. Next, clean the silicon probes with distilled water. Under the microscope, paint the probes with a fluorescent marker for marking the positions of the probes in the histological analysis. This procedure should always be done under a microscope to make sure the probes are not being damaged or bent. After that, shave the fur along the incision site. Then, apply ointment to both eyes. Administer 5 mg per milliliter of dexamethasone subcutaneously to reduce brain inflammation. At this point, fix the animal in the stereotaxic frame. Next, prepare the surgical site antiseptically using chlorhexidine soap and ethanol. Before making the incision, administer lidocaine and epinephrine subcutaneously at the incision site for local anesthesia and to reduce the potential risk of bleeding. Then, make a 2 cm incision along the scalp midline and remove the skin to expose the skull. Clear the dorsal scalp from the fascia by blunt dissection and control bleeding using bone wax. Next, drill 2 holes in the occipital bone of the skull. Attach the small screws to the holes for ground and reference electrodes. The reference screw must be in contact with the dura. After that, make 2 1 to 2 mm diameter craniotomies in the coordinates corresponding to the forelimb somatosensory cortices. Use compressed air to remove the bone dust as needed during craniotomy. To prevent the skull warming up from drilling, apply drops of saline solution. Then, build a small barrier of acrylic resin surrounding the 2 craniotomies in order to keep PBS on top of the exposed dura and to prevent it from drying. Apply PBS constantly during the whole experiment. Next, use 2 30.5 gauge needles with bent tips to remove the dura mater of both craniotomies. At this point, place the tips of the silicon probes just in contact with the pia. Then, slowly introduce the silicon probes, making sure that their shanks can be inserted into the brain without resistance or bending. Carefully lower the probes until they reach the desired recording areas. If needed, add drops of PBS on the top of the craniotomies to prevent the tissue from drying. Shown here is a representative electrophysiological recording of the local field potential for 500 milliseconds from one tetrode. An example of the spike waveform is in the insert. Shown here are examples of the 2-dimensional views of unit clusters obtained from one tetrode. Each cluster represents the spikes from a single neuron. The averaged spike waveforms from the representative units at each of the 4 channels from a single tetrode are shown here. The waveform energy of the recorded spikes over time is plotted in this graph. Note that the relatively constant energy value for each unit indicates stable recording over the displayed period.
After watching this video, you should be able to perform silicon probe recordings in multiple brain areas from more than 100 neurons simultaneously. This is achieved by completing the following steps: anesthetizing the animal, placing the reference and ground screws, drilling the craniotomies, removing dura, and finally inserting the probes in the desired recording area.
Recommendations. When inserting the probes in brain tissue, it is possible to cause considerable damage. This can result in a low quality of recorded units. To avoid this problem, we recommend the following. 1. Introduce the probes at a certain degree angle, recommended to use a 10-degree angle. By doing this, dendritic damage of the recorded neurons can be reduced. 2. After the probes are inserted in the brain tissue, they are initially lowered at a faster rate, approximately 50 to 100 microns per 10 to 30 seconds, until they get closer to the desired area of recording. When the probes are about 200 microns from the target area, the position is adjusted more slowly, approximately 10 microns every 2 or 3 minutes. Recommendations. When the probes are in the designated area and considerable activity is detected, distinctive spikes in the majority of the channels, it is recommended to wait approximately 30 minutes before starting a recording. This will allow brain tissue to mechanically stabilize after the insertion of probes and ensure a more stable recording. Occasionally, it is possible to see brain pulsation that can significantly reduce the quality of a recording. A small craniotomy, just enough space to fit the probe, can reduce brain pulsation in a recording area. Recommendations. Silicon probes can have a variety of shapes and recording site configurations. For example, they can vary in the number of shanks, the length and thickness of shanks, and in the arrangement of recording sites, e.g. tetrode versus linear configuration. See www.neuronexustage.com. The choice of probe used with a specific configuration depends on the scientific question needing to be answered. For example, if the objective is to record populations of neurons in multiple locations in one specific layer, as presented in these recordings, the best choice is to use a probe with 8 shanks and a tetrode configuration. Thank you for watching. Subscribe if it is useful.